Thanks, Mike, for that very kind and slightly over-exaggerated introduction. Thank you also for the organising committee for inviting me to give a plenary lecture on the genomics of platelet disorders. Here are my affiliations, and here are my disclosures, none of which are directly relevant to this presentation. So I'd like to put this talk into context first by showing some registry data from the United Kingdom, and actually the message is the same from other national registries. In the UK, we have about 60 million people and nearly 27,000 people registered with bleeding disorders. And as expected, this list is dominated by coagulation factor deficiency disorders, von Willebrand disease, Hemophilia A. If we consider inherited platelet disorders, then in the UK we have a total of nearly 2,200 patients, but almost certainly this is a significant underestimate of the true numbers. Even this number puts it at a similar level to factor XI deficiency or Hemophilia B. The other important difference between this group of inherited platelet disorders and the other diagnoses on this list is, of course, with one or two exceptions, the coagulation factor deficiencies at the top of the table are simple monogenic disorders caused by pathogenic variants in single genes. By contrast, inherited platelet disorders are a heterogeneous group with complex genetics. This graphic summarizes a currently up-to-date list of approximately 50 genes that have been previously linked to inherited platelet disorders. And the important message from this slide is that genetic platelet disease can affect platelets from very early in megakaryocyte development right the way up to mature platelets. So genetic disorders in this group here affect the function of mature platelets. So these are classic platelet function disorders. Genetic disorders in this area here in megakaryocyte differentiation typically manifest in platelet number disorders. There's a further group in this region here of the slide that impair formation of platelet organelles, which also manifest as platelet function disorders. Of course, this distinction into platelet function versus platelet number disorders is not absolute, and there are some important disorders here where there are abnormality in both number and function, perhaps best illustrated by Bernard-Soulier syndrome caused by gene defects here. So this kind of subclassification of inherited platelet disorders is very interesting scientifically, but how does it help clinically our patients? What I'd like to propose then is that working out which gene is defective in our patient with platelet disorders is important for precision diagnosis, just like coagulation factor disorders. And also, just like coagulation factor disorders, precision diagnosis has got all sorts of clinical benefits for our patients, and I've listed some of those on this slide. And just to convince you further, I'll just illustrate with an example from the clinic of just one of these about prognosis. Consider here then an index case presenting for evaluation in the hemostasis clinic who's pregnant and has been noted to have mild thrombocytopenia but no other clinical abnormalities. This patient's got no other features suggestive of an acquired thrombocytopenia, but does have a family history of thrombocytopenia in a father and a paternal uncle, so suggestive of a dominant platelet number disorder, heritable thrombocytopenia. This is quite a common presentation, and here are two different blood films from two different women in the clinic. The top one here shows mild thrombocytopenia with normal-sized platelets. The bottom film here shows mild thrombocytopenia with normal-sized platelets. They're essentially the same film, and we can't go any further at this stage without molecular diagnosis. The top film comes from a lady who's subsequently been shown to have a pathogenic variant in CYCS, so thrombocytopenia 4. So this is a very benign chronic thrombocytopenia with no other clinical manifestations and associated with relatively low bleeding risk. Contrast this with the film below. This is from a lady with a pathogenic variant in RUNX1, so almost certainly associated with a higher bleeding risk than CYCS, and at least in some families with some variants associated with a very penetrant increased risk of myeloid malignancy, so familial thrombocytopenia with a propensity for acute myeloid leukemia. So in this simple example, genetic diagnosis, precision diagnosis, has allowed us to give radically different estimates of prognosis for these two families. What I'd like to do in this presentation is explain, with the help of some case studies, how understanding the genomics of platelet disorders helps us improve precision diagnosis. I want to cover three separate areas: our current tried and tested approach, that's candidate gene approaches. I'll move on to talk about candidate gene panels and exome sequencing. And then finally, at the end of the talk, touch on whole genome sequencing and how we can use this for gene discovery. And I'll finish up finally with some future directions. So let's go back to the clinic to discuss how we do candidate gene approaches. So here's one of my patients who, as you can see, has presented for evaluation at a very early age with some abnormal mucocutaneous bleeding. Like most centers faced with a patient like this, we would do some coagulation factor assays and study platelets by flow cytometry. And here we can see the answer. This is flow cytometry looking for surface expression of CD61. That's the beta-3 integrin. And as we can see, compared to an age-matched control, our patient is showing markedly reduced expression of this integrin, consistent with a diagnosis of Glanzmann's thrombocythemia. Now, we all know that Glanzmann's is a very well-characterized platelet function disorder and is caused by defects in just two genes. So if we want to offer this family genetic diagnosis, then we've only got two genes to look at. Here's one of them. This is ITGB3. And the conventional approach with this kind of simple case with a small number of candidate genes is PCR of the coding regions of the gene exon by exon. We would then subject this PCR amplicon to a technique such as chain termination sequencing, that's Sanger sequencing, and compare the sequence here with a reference sequence looking for variations. And here we can see an example of a heterozygous single nucleotide variation in this trace. Finally, we then compare variations that we've annotated against a transcript for ITGB3 with a disease-specific mutation database to see whether this has previously been reported in another family of Glanzmann's thrombocythemia. So in this simple example of a recessive platelet function disorder, if we find two changes like this in the same gene, or possibly the same change twice, indicating a homozygous change, that's been previously described in a sufficient number of families that are unrelated with the same phenotype, and it's a rare variant, then we've got good evidence this is the causal gene variation and a presto genetic diagnosis. Here's a second example that illustrates that this is often not so easy. Here the phenotype's different. This is an adult presenting here with mild surgical bleeding, and by light transmission aggregation compared to the control C, our patient is showing diminished aggregation responses to ADP, but actually comparable responses in control with other agonists studying the collagen and thrombin receptor. So using the same approach as Glanzmann's, it would be natural to think that the two candidate genes for someone with this phenotype would be our two platelet ADP receptors. And actually, these genes are very small and easy to study. However, there's an important difference between our phenotypic test here and the phenotypic test for Glanzmann's thrombocythemia. Tests like light transmission aggregation offer much less diagnostic specificity. And in fact, if we have to consider all the things that can go wrong in a platelet for the response to ADP to be abnormal, we have to consider not just the receptors, but the whole signaling pathway. And this is just a simplified version, but even here, it's easy to see that if we want to select a candidate gene for defective ADP response, we have to consider an awful lot more proteins than just two. So this kind of scale of sequencing required for genetic diagnosis is clearly beyond the scope and cost effectiveness of Sanger sequencing. We have to find a way of analyzing simultaneously lots of different genes for diagnosis in our patients. So Johannes Oldenburg yesterday gave us a very nice summary of current next-gen sequencing technology, and I'll just recap with one slide, just one example of the several available technologies available. What next-gen sequencing technologies have all got in common is that they enable simultaneous sequence analysis of lots of different genes at the same time. In this particular example, the first step is to design a library of mRNA baits that cover the region of the genome required to be sequenced. In this example, each bait is represented by a black rectangle, about 200 base pairs long, and here we can see that we require 6 different mRNA baits to cover just one coding exon of a target platelet gene. The principle of the analysis is that genomic DNA samples from our patients are fragmented, mixed with these baits, and then labels on the end of each bait are used to pull out the targeted sequence from the DNA mixture. There's then an amplification step followed by simultaneous sequencing of all the targeted regions. The output from this then is lots of different individual sequence reads, which are analyzed bioinformatically and compared to a reference sequence. From this data set, we can then pull out single nucleotide variations and also now at least small insertion-deletion variants. This is a flexible technology because we can select exactly which regions of the genome we'd like to sequence. So we may want to select specific panels of genes, so that's coding sequence in gene panels. We may wish to cover all coding genes in the genome, so that's whole exome sequencing. Or we may wish to cover the whole lot, all 3 million bases, so that's whole genome sequencing. What I'd like to do is show an example first of how we're using gene panel sequencing as a diagnostic test. There are several different gene panels for bleeding disorders, but I'll just describe just one. This is the thrombogenomics panel, which is currently at an advanced evaluation stage within the UK NHS. So the thrombogenomics panel then is a targeted gene panel for bleeding and platelet disorders, but in its current form covers 78 genes previously linked to human bleeding, platelet, or thrombotic disorders. Specifically, it covers the coding regions of these genes and selected regulatory elements, and in total just over 1 million base pairs. The intended use of this panel is for diagnostic evaluation, precision genetic diagnosis of people with already characterized bleeding or platelet disorders, in which there's already a phenotypic clue as to which pathway or even which protein is likely to harbor a causal gene defect. The crucial property of this, though, is that irrespective of the phenotype of the patient, all 78 genes, which include about 30 genes here for platelet disorders, undergo the same automated routine analysis. The first step in the analysis pathway is that the sequence reads from the next-gen sequencing for all these genes is annotated relative to the transcripts of these genes that are expressed in megakaryocytes or platelets or hepatocytes. Common variants are then excluded from the analysis, and then annotated gene defects are considered in an MDT meeting. This is an example of the curated output from just one patient from my clinic, actually, and we can see already that even in just 78 genes analyzed in this panel in our patients, we've got 20 different annotated variants indicated here by the numbers in these different gene boxes. So the purpose of the MDT in programs like this is to consider which of those variants is causal in the patient. And of course, the things that will be considered will be the clinical and laboratory phenotype of the patient, whether that variant's likely to be pathogenic within the transcript, and whether that pathogenic variant is likely to explain the phenotype of the patient. So in this particular example, in which there's a patient presenting with oculocutaneous albinism, who has absent dense granules on electron microscopy, and who has a homozygous stop-gain variant in the HPS3 gene, it's fairly straightforward to suggest that the causal variant is in HPS3, indicating a diagnosis of Holansky-Pudlak syndrome type 3. So this program clearly works on an individual patient basis, but if we're going to use it as a routine diagnostic test in the clinic, we need more information from larger patient groups about its diagnostic accuracy. And again, I'm presenting some data here from the thrombogenomics program from a paper which describes the evaluation of the next-gen panel in 300 cases by parallel testing DNA samples using the thrombogenomics panel by next-gen sequencing and also our gold standard sequencing assay, that's Sanger sequencing. And what these results show us is that if you use DNA samples from patients with bleeding disorders who've already got a known causal variant, the panel and a blinded MDT group will identify the causal variant that's correct in 100% of the cases. If you use a patient group in which the referring clinician suspects a specific disorder, but genetic diagnosis hasn't been done before, then the panel and the MDT group get the right answer, a causal variant, in nearly 92% of cases. So this is very good diagnostic performance. The panel is also very cost effective. And so data like this and validation from the other gene panels for hemostasis disorders internationally, I think together are telling us that gene panels are very, very likely to be a cost effective and clinically useful molecular diagnosis tool going forwards, at least for some patients. Some patients, but not all patients, because there's also a flaw in this plan indicated on this slide, and that's the bottom row of the table. The thrombogenomics program also evaluated a group of patients with bleeding and platelet disorder, but of unknown etiology. So these are people with bleeding disorders with normal coagulation factor levels, or with platelet disorders that don't fit phenotypically very well with already described disorders. When this group of patients were tested using our panel of 78 platelet and bleeding genes, then only 11% ended up having a causal variant that was confirmed by Sanger sequencing. Now that 11% isn't because somehow the next-gen panel failed to detect variants. It was because this group of patients didn't have a variant in the 78 panel of genes. So if they haven't got a variant in the 78 panel known bleeding and platelet disorder genes, where is it? Well, it must be somewhere in the genome. It's very likely to be in an exon of a coding gene, which is amenable to next-gen sequencing, whole exome sequencing, or maybe it's lurking in an intron or in a regulatory sequence, in which case it's amenable, in principle, to whole genome sequencing. However, initiatives like the Human Genome Project have now defined the architecture of the human genome in much more detail and important food for thought about how we're going to interpret sequence data. Recent estimates of the human genome suggest that, in contrast to the 78 known bleeding and platelet disorder genes, there's a total number probably of over 20,000 individual protein codon genes. If you consider all the possible different transcripts those genes could produce, then we have nearly 170,000 different transcripts, many of which tissue-specific. And perhaps the most startling statistic of all is that of all the human genomes that have been sequenced to date, a total number of more than 62 million individual sequence variations have been identified in human beings. If we look at data from initiatives like the NHLBI Exome Sequencing Project, so this is the excellent EXACT database, or from the UK 1,000 Genomes Project, we get the same important message. If we look at individual patients, each individual who undergoes just exome sequencing has an average of 14,000 different single nucleotide variations across all coding genes. Many of those, of course, aren't pathogenic, but if you computationally predict the proportion of those variants which confer loss of function to the gene that they're located in, then each one of us in this room and each one of our patients has, on average, about 100 predicted loss of function genotypes in our entire genome. So the challenge then of applying whole exome sequencing and whole genome sequencing to genetic diagnosis in our patients with inherited platelet disorders is not now sequencing 3 billion nucleotides. It's actually in resolving the single causal variant amongst this background of very high inter-individual variability. I want to return to the clinic now for a further example about a way of getting around this significant technical headache. So the final case I'd like to describe in the presentation is a 35-year-old woman who's referred to a clinic in Bristol for evaluation of persistent obstetric bleeding. Her phenotypic tests show that she has mild thrombocytopenia. She also has enlarged platelets, so that's macrothrombocytopenia. Significantly, she also has sensorineural hearing loss. It's bilateral, it's profound, requiring hearing aids, and developed at least as early as infancy. What's important about this illustrative family is that she has 4 children. 2 had both macrothrombocytopenia and hearing loss, and 2 had neither. So this is suggestive of a syndromic association between these phenotypes and suggestive of a monogenic disorder tracking it as an autosomal dominant trait. Many in the audience are going to be familiar with this association between hearing loss and macrothrombocytopenia in patients with variants in MYH9. So that's MYH9-related disorder, in which at least some people have this syndromic association. So the initial evaluation of this family was to put DNA samples through the thrombogenomics panel, specifically looking at MYH9 as a single candidate gene. Well, actually, no variant identified, and no causal variant identified in any other bleeding or platelet disorder genes. So this was a new gene discovery project, and the next step up was to put DNA samples from the index case through whole genome sequencing. The crucial message from this family, though, is that we didn't just do whole genome sequencing on the index case or family members. We put the genome sequencing data into a very large international program. And that's the Bridge BBD project. Again, there are several international genomic projects in bleeding and platelet disorders. I've highlighted this one because it's probably the largest. It's truly international and has to date recruited nearly 1,100 individual cases with uncharacterized bleeding and platelet disorders, almost all thrombogenomics panel negative, so with potentially new causal genes. And each one of those 1,100 cases has undergone at least whole exome sequencing, and in most cases, whole genome sequencing. The other crucial characteristic of the Bridge BBD study is that phenotype is considered to interrogate whole genome sequencing results, but it's phenotype that is expressed in human phenome ontology terms. So for those of you who aren't familiar with this system, this is a system of naming phenotypes using semantic terms in threads, which range from general terms right the way down to very specific phenotype terms. So here's an example of human phenotype ontology terms referring to just one patient actually with grey platelet syndrome. So they have, for example, a disorder of the number of alpha granules, which is a kind of disorder of alpha granules, which is a kind of disorder of platelet morphology, which is a kind of disorder of blood and blood-forming tissue. They've also got a big spleen, so that's splenomegaly, a kind of disorder of the spleen, a kind of disorder of abnormal organs. What this achieves, it means that even complex phenotypes can be expressed mathematically in strings of code, which means they can then be compared to assess how similar they are to the phenotypes of other people with bleeding and platelet disorders and other syndromic features elsewhere in the cohort. Here's an example of how it works in our pedigree. So on the left here, we see an HPO node diagram expressing mathematically the phenotype of our patient. And we can see here a big node for sensorineural hearing impairment and another smaller node for thrombocytopenia. The general principle of the analysis in the Bridge BBD study is that mathematically expressed phenotypes are then compared between individuals across the cohort to assess the degree of similarity. If 2 unrelated index cases have got mathematically very similar phenotypes, particularly if that's an unusual phenotype within the cohort, then there's a very good chance they've got a defect in the same gene or at least gene pathway. The statistical genetics colleagues on the program can go one step further, and they can calculate the probability of an association. So this is an approach called similarity regression, and what they've done here is they've considered all of the high impact variants in all exons of all genes in all members of the Bridge BPD cohort, 1,100 people. And they've asked the question, what's the probability of an association between a variant in any given gene is true between individuals who share this phenotype? What we can see here is there's one very clear winner. There's a strong hit with a high probability of causal association in a gene called DIAF1. And lo and behold, if we sequence DIAF1 by next-gen sequencing or by Sanger sequencing in our index case, we find they've got a high-impact variant here in the penultimate exon of the gene, predicting a substitution of arginine 1213 with a premature stop codon, here crucially in a regulatory region of the protein. So here, whole genome sequencing and statistical genetics techniques have given us a very good candidate causal gene for this particular phenotype in this particular patient. Does this constitute proof of a new human genetic disorder? Well, not quite. We have to do several other things now to prove that this is a truly causal association. Perhaps the most obvious one is to test whether this variant segregates within our index cases family with the phenotype. Well, it does. The variant is present in heterozygous form in our index case and also 2 of the affected 4 children, but not present in the 2 unaffected children. What about the other families in the Bridge BBD cohort which were found statistically to have a similar phenotype? Have they also got a variant in DIAF1? Well, only one other family had a mathematically similar phenotype to this index family. And here they are, and here's the index case. And this is a highly specific phenotype within the cohort. This entire family had autosomal dominant macrothrombocytopenia and sensory neural hearing loss. And not only did they have a variant in the same gene, DIAF1, they had an identical variant to the initial index case family. So there's good segregation here in more than a dozen individuals between this specific variant and this very specific phenotype. Is this a biologically credible gene change? Well, the DIAF1 gene encodes a protein called diaphanous-1, which we know is a cytoskeletal regulator. We can see here the DIAF1 protein polymerizing an actin filament, and it does something very similar to microtubules. So this is biologically plausible because we know that microtubules and actin are both important for proplatelet formation during megakaryopoiesis. And we know specifically that DIAF1 protein is an important negative regulator of protein platelet production. We also know that DIAF1 and the cytoskeleton is important for hair cell development. And of course, hair cells are the origin of stereocilia in the inner ear and essential for hearing function. So there's also a good biological fit here with the hearing loss phenotype. Finally, because this is a new gene discovery, we always seek experimental proof that the causal variant identified in our human patients is reproducible in other experimental systems. So here we've expressed the variant DIAF1 protein in a human cell line to show that it exerts the same phenotype on the cytoskeleton as observed in cells from our patient. And we've tested the effects of knockdown of the DIAF1 protein in an animal model, in this case zebrafish, to show that it exerts a hematopoietic phenotype and also a hearing and balance phenotype. So just to summarize this gene discovery case, we've got a patient who had a candidate gene for a specific phenotype, but no causal variants in that candidate gene, MYH9. Therefore, they went into a gene discovery program with whole genome sequencing. By combining with a very large cohort of patients and using human phenome ontology terms, we were able to identify a causal variant that we showed segregation, biological plausibility, and experimental confirmation was very likely to be the causal gene. Since the discovery, further families have now come forward with similar phenotype and variants in the same gene. And together, this body of evidence allows us to promote the DIAF1 gene from being a candidate plausible gene to a tier 1 gene, a gene that's very likely to be linked to human platelet disorders. And DIAF1 has now been included in the thrombogenomics panel as a routine diagnostic gene for patients with heritable thrombocytopenia. In fact, if we look at all the genes that have been added to the thrombogenomics panel within the last 5 years, including DIAF1, almost all of these have been enabled by genomics, in particular by whole exome or whole genome sequencing, to be added to a routine diagnostic test. In terms of future direction, everything we've talked about in the presentation so far has been about causal variants within the coding regions of genes, specifically in the exons. What we now understand about the architecture of the human genome is actually 98% of our more than 3 billion nucleotides don't code for protein. They're in the non-coding space. But that's no longer viewed as just junk DNA. We now know that this region contains important regulatory sequences, in particular proximal regulators, gene promoters, and long-range regulators, including elements like enhancers, repressors, and insulators, both of which interact to tightly regulate the expression of individual genes, including platelet genes. I'd like to present just 2 slides of unpublished data from the Cambridge Blueprint Consortium that just gives a flavor of what genomics is doing now for human platelet disorders. This is epigenetic mapping of a region of chromosome 14 that contains the actinin-1 gene. And actinin-1 has been previously linked to human platelet disease because loss of function variants in this gene appear now to be a common cause of non-syndromic autosomal dominant heritable thrombocytopenia. And what we're looking at here is biochemical modifications of the region of non-coding chromosome 14 that's just downstream and within the actinin-1 gene. What this is telling us is these peaks indicate histone acetylation, and these peaks in green here indicate a particular kind of cytosine methylation, which are both epigenetic markers indicative of key regulatory elements. And we can see immediately there's a consistent peak here some way downstream of the actinin-1 gene. Could this be a potential regulatory region for actinin-1? Well, we can also use genomics now to study whether there's a DNA interaction between this region and the actinin-1 gene. So this is chromosome immunoprecipitation sequencing, ChIP-seq, again in this circular graphic indicating the region of chromosome 14 that's in immediate proximity to the actinin-1 gene here. Here's our putative enhancer downstream from the actinin-1 gene, and these curved lines indicate experimentally proven DNA-DNA interactions. And what this tells us immediately is that almost all of the DNA interactions that feed back into the actinin-1 gene come from this enhancer and not from any other places in chromosome 14. So going forward, if we're having to look at new genetic regions, particularly in the non-coding space, that could contribute to human platelet disease or even could be causal, I think it's in regions like this we should be looking. I'll finish with a summary slide. I hope in my presentation I've conveyed to everybody that genomics has already impacted on our clinical diagnostic practice for inherited platelet disorders, specifically through the launch of targeted gene panels that include known human genes for human platelet disorders. I've also conveyed that going forward, whole genome sequencing is now a proven tool for gene discovery in inherited platelet disorders. However, it comes with some cautions. It's necessary to perform whole genome sequencing within large study cohorts. It's necessary to provide detailed phenotyping using models such as human phenome ontology to enable resolution of causal variants. And it's also necessary to deploy statistical genetic techniques to be confident about resolving the single causal variant on a background of high variability between individuals. My main message from the presentation is that whole genome sequencing and similar genomics approaches is undoubtedly entering clinical practice as a diagnostic tool, and in the NHS has already been launched as routine for rare disorders, including platelet disorders, through the UK 100,000 Genomes project. Thank you for your attention.